Good afternoon, everybody, and welcome also to those of you watching online. Can you hear me okay? Can you hear me okay? Is this on? Oh, good. Welcome and thank you for coming to this week's seminar. If you would like to tweet, the hashtag is OMS10. But please could I remind you to put your devices on silent. And for those of you watching online, I will try to take questions via Twitter if we have the time. Today's speaker is Professor Angela McLean. She is one of the people who we think of as one of our original professors. The Institute for Emerging Infections was one of the first programmes that the Oxford Martin School funded, and Angela is now also director of a new programme looking at collective responsibility for infectious disease. So looking at the human factor that's involved in controlling infectious disease. Angela has a great long list of accolades to her name. She is a Royal Society Fellow. She's a Fellow of All Souls College here at Oxford. So she is one of our most eminent professors. And today she will be taking a look at some of the work that the programme has dealt with over the years and hopes to give you an insight into that. So I hope you'll all join me in welcoming Angela.
So I've been told very firmly that I must stand here and not move because of the microphone. So if I move over there, you all have to say, go back, okay? Because I will forget. So first of all, let me start by saying a very sincere thank you for the invitation to be here today. I very specifically made a title called Understanding Emerging Infections, because what I'm going to talk about today is really very firmly focused on trying to understand particular things about the evolution of one emerging infection, and that is HIV. But I'm going to start by putting it into the slightly broader context of emerging infections in general. Ever since I was a grown-up, emerging infections have been an issue. They've been in the news. So, in fact, as I was finishing school, was the time of the very last, what we might call wild smallpox cases. Those were in 1977. Around about the same time, there were a couple of the very earliest Ebola outbreaks. But really, back then, there was a feeling that infectious diseases were something that were under control. But that didn't really last very long, because in 1981, the first cases of AIDS were described. They certainly weren't the first cases that existed, but it was for the first time the disease caused by HIV was described. And I put it up there as in 1983, because that was the year that the causative virus was discovered. So this is a list of just some of the emerging infectious diseases that have emerged in whatever that is. I guess that's about the last 30 years. And they really fall into, I would say, 5 categories. So the first is if you have the new identification of something that's been around for a long time. Let's see if this works. So, for example, hepatitis C virus has been around for hundreds and hundreds of years, but was identified in 1989. Or an alternative version of that is when you get a new association between a pathogen that you know about and a disease that you've known about for a while, and you realise that pathogen is actually a causative factor for that disease. So that's sort of type number 1. The second kind of emerging infectious disease is when you get a new infectious disease in a place where you didn't have it before. So an example of that on this list would be chikungunya. That's a vector-borne viral infection, an outbreak in Italy in 2007. Third example, and the ones I suppose I'm really pretty interested in, would be newly evolved forms of known infections. So influenza would fall into that class, and so would the drug-resistant pathogens that are becoming such a problem. Then there's the genuinely new things, so things that you just didn't have before, and that, as far as we can tell, and we usually can tell, have not been circulating in humans before, and HIV is one of those. It wasn't new at that time, but we know that it crossed over into humans during that century, basically. And others like that are this strange thing called lujovirus, that I'm not going to talk about today. MERS coronavirus is another one of those. And then finally, and perhaps in some ways I think sort of most depressingly, there's what we call re-emergence. And that's where something that we thought was under control comes back again. And I would say really the most depressing set of those are the re-emergent infections that ought to be controlled by childhood vaccination. And you see that on here with polio re-emergence in 2004 in Nigeria. And that's a whole interesting story about why people in Nigeria started to refuse polio vaccination and how that led, well, fundamentally to outbreaks all over the world as polio control failed, first in Nigeria and then in countries where cases were exported from Nigeria. Again, that's not the one I'm going to tell you today. I forgot to bring it with me. Every month I get sent a two-sided piece of paper from Public Health England that's describing events like these. It's what our government does for what's called horizon scanning. So we're trying to keep an eye on what's going on around the world that might cause a problem for public health here in England. And there's certainly more than one of those every year. So the one that I've got sat on my desk next door for September has at the front of it, actually the top article is about dengue. It's about a series of 6 cases of dengue, which we think of as a tropical disease, acquired in southern France. Only the second article is Ebola now, but of course, for more than a year, the most important worry has been about Ebola in West Africa. So don't get the idea that these things only happen once a year. Here's a digest from one year I happened to be following for an article I wrote. So from June 2013 to May 2014, I just picked one example from each month of the sorts of things that were going on. And again, they fall into the same group as before, the same 5 groups as before. So you could have a new disease or a new association between a disease and the known pathogen. So there's one here, a report from Portugal of a putative association between hepatitis E virus and Guillain-Barré syndrome. A new place for a known disease. This is one of the most important things that happened during that year. That was the first cases of this virus, chikungunya, in the Caribbean. We think of chikungunya as a virus of the tropical old world. I pointed to you to there was a small outbreak in Italy. This has since then developed into an absolutely enormous outbreak. So this is a viral infection transmitted by mosquitoes, used to be confined to this part of the world, now spreading through the Americas for the first time ever. Newly evolved versions of things we know about, probably the biggest problem we have with infectious disease, antimicrobial resistance. I've put that down as April 2014 because in that month the WHO published a huge report trying to summarize how much we know about antimicrobial resistance around the world. And then the brand new things, MERS coronavirus in Saudi Arabia was getting reported in July 2013. And then back again for the 5th one to the things I find most sad, and again it's polio in this case. This was polio being exported from Cameroon, Syria and Pakistan, and this was actually declared as an infectious disease emergency of international concern before the Ebola virus was. So that was because it was so unusual at the moment for polio to be being exported across borders from one country to another. I do have time actually, so I'll tell you. I came back to teach here in Oxford in the year 2000, and I teach infectious disease biology to undergraduate biologists. And that year I wrote a lecture, and the title of that lecture was, "Is this the year that polio will be eradicated?" And that's a really sad story actually, because things were better then. There was genuine hope then. I mean, fundamentally, through most of my adult life until then, there had been genuine hope that polio was going to be eradicated. And I think if one is realistic, one will soon have to say that perhaps that hope is fading. And there's really no good reason for that. We have a cheap vaccine that is mostly very effective. And the reasons that we can't use that vaccine to eradicate polio are really political and social, not biological. Anyway, so in this year that I was tracking the re-emergence event, the most important re-emergence event, was the re-emergence of polio in Africa and in the Middle East. And this was, of course, also the year that Ebola virus emerged for the first time in West Africa. Actually, Ebola's in the news again today, but for a happy reason, which is that the Scottish nurse who had this very strange resurgence of her own Ebola infection that made her so very ill has in fact been sent home today. She's well, she's not infectious, she's gone back to Scotland, and that's from today's Guardian. And the other really good news about Ebola this week is that on Saturday, Sierra Leone was declared Ebola-free. So Liberia has been Ebola-free for a while, and in fact there were no new cases of Ebola in Guinea last week. So the current total for that West African outbreak is something over 28,000 cases and something over 11,000 deaths. So with emerging infections, we very often ask questions about their evolution, and Ebola's no different. But I'm going to show you a slide about the sorts of questions that get asked about Ebola evolution that I hope will persuade you that to the things I'm interested in, it is not a good model organism. So if you want to talk about, so there's been a lot of discussion in the popular media about might Ebola become airborne. So at the moment, Ebola is not airborne. It's something that you catch by rather close contact with people, and it's really bad if you catch it, but it's not that easy to catch. Over a year ago, Mike Osterholm wrote an op-ed piece in the New York Times entitled "What We're Afraid to Say About Ebola," in which he seriously raised the possibility that Ebola might become airborne. And he actually got quite a lot of kickback from other virologists saying, listen, there's a lot of things to worry about, and that actually isn't one of them. Including from this person here, Vincenzo Raccanello, who I think is an absolutely fantastic virologist who writes this blog. We'll come back to this in a moment. So this op-ed was written in the New York Times, caused a fairly strong backlash, and that seemed to be the end of it, until February of this year, when the same group of authors wrote a peer-reviewed article that was mostly a pretty straightforward review of the biology of Ebola, and again raised the issue that Ebola might become airborne. And that was picked up very widely in the international press. Our own lovely Daily Mail health pages covered it, the Washington Post covered it. And again, there was really quite a strong pushback. CDC issued this statement, why Ebola is not likely to become airborne. My favourite virology blogger wrote this article called "Ebola virus will not become a respiratory pathogen." And you can see what's going on here is a certain amount, sort of more or less like Punch and Judy. Yes, it will, no, it won't. Yes, it will, no, it won't. And that's because this is an extremely difficult thing to study in Ebola. People who are infectious are extremely ill, very difficult to follow, can be done, but this particular question is really barely amenable to experimental work. And what you end up with is people raising scary possibilities, and the only sensible answer one can give is, well, by analogy with everything else we know, we really think this is very unlikely, but at the end of the day, that's not really the kind of evidence we'd like to gather. My ferret picture is to remind me to tell you that it's not just for Ebola that people ask these sorts of questions. So we're often very interested in influenza infections, in questions about how it might evolve. And in particular, we'd like to know if new forms of influenza that we know about might become more virulent, i.e. make people more sick if they catch them, or if they might become more transmissible, or indeed if they might become transmissible via new routes. So the reason I got the picture of the ferret is that these very controversial experiments done with ferrets in Holland, asking if it's possible to create a bird flu that is transmitted by air, so between ferrets who have no direct contact with each other. So there's a sort of series of questions that we tend to ask again and again about the evolution of emerging viruses, in particular to do with how sick they make you, how transmissible they are, and well, those two really, how they're transmitted. So one of the reasons we work on HIV is that all those are much more amenable to what I would call proper direct study during HIV infections. There's a large there are large population studies done of viral diversity, so you can collect your own, and you can also use data that's been collected and deposited in huge publicly available databases about virus sequences. There are also models in vitro, so you can grow this virus in a test tube and measure its properties there. And I believe it's an excellent model for asking about the evolution of a new emerging infectious disease in humans as it spreads through humans' populations for the first time. And it's, of course, also by far the most important of the emerging infectious diseases that we've been confronted with. So to back that up, let me show you, these are the most recent UNAIDS statistics, so they're published this year and cover 2014. So in 2014, there were nearly 37 million people living with HIV. During 2014, there were 2 million new infections, and during 2014, there were 1.2 million deaths. So just to put that into perspective, do you remember we talked about there having been 28,000 Ebola infections during the whole of the West African Ebola epidemics? Well, we'd have had that in just slightly more than 5 days during 2014 with HIV. And if we think about deaths, there were 11,000 deaths during the entire period of the Ebola epidemic, which, touch wood, is very nearly over. And for HIV during 2014, we would have had 11,000 deaths in about 3.5 days. This is a huge health problem. But when you look at these figures, I think, in a way, it sort of reveals whether you're a glass half empty or a glass half full person. Because actually, these figures are terrifically good news in some sense. But in order to get that perspective, you really have to look at how we got here. So if we go back through time, these are also UNAIDS statistics, but covering from 1990 to 2014. So remember, this was an infection and a disease that were both unknown in 1980. Remember, the first cases were described in 1981. And these lines don't go back to there because nobody's prepared to make the rather heroic assumptions that would be required to fill in that data. That data's just missing. But what we have here is the number of people living with HIV. So there it is at 37 million on this axis here at the end of 2014. And what you see is an inflection in this curve. This curve is not going up as fast as it used to. And without intervention, we would have expected this curve to be rising exponentially. So it's not just that this straight line didn't go on going up, it didn't get faster either. And the reason that this has got so much better is this, that the number of new infections plateaued round about there and has started to fall, and the number of new deaths, the number of AIDS-related deaths, rather later, also plateaued and started to fall. And of course, the rate at which this goes up each year is simply the difference between how many people got newly infected that year, in green, and how many people sadly died that year. And you can see that the reason this is going much more slowly now is that the difference between these two is now much smaller than it used to be. And I think there are two explanations for that. The one that's absolutely clear is the provision across the world of extremely effective drugs that treat people who have this infection, prolong their lives, that's one of the main reasons why the number of deaths is falling, but also make them certainly very much less infectious. Many people would say not infectious at all. So this has been a fantastic example of how doing things that are good for individuals, namely treating their own personal HIV infection, has also been tremendously beneficial for the population, because those people are no longer infectious to the people around them. So what I want to do now is pose 4 questions about the evolution of HIV. Now, the following 4 questions. So is HIV evolving to become less virulent? How fast does HIV evolve within infected people? If we look at patterns of HIV evolution, are they consistent across sub-epidemics? So although we think of this as one global pandemic, we could also look at it as a bunch of sub-epidemics happening in different parts of the world. And then finally, when people are on this therapy that seems to, that does them so much good and makes them virtually uninfectious to other people, is HIV nevertheless still evolving inside those people? So with the first question, why ask this question? So we need to start off, first of all, there is a received wisdom that says that, well, no pathogen would ever want to kill its host, because a pathogen's much better off in a host who's alive running around infecting other people. That's quite a strong received wisdom, and completely fails to capture the idea that a pathogen has to do two things, basically. One, ideally, yes, keep its host alive and transmitting as long as possible, but it also has to get out of its host into another person. And much of what makes us sick is the process of the pathogen doing that. So this is what we call a trade-off hypothesis, where actually it's not, we would not expect, so under the trade-off hypothesis, we would not expect pathogens to become avirulent. We would expect them to evolve to a place where the balance between keeping your host healthy enough to run around transmitting other people, but having enough virus in that host that they do indeed transmit when they have a contact is about right. And so I think it is very important to understand that the received wisdom that says, oh, a good pathogen is an avirulent pathogen is really very naive. So the trade-off hypothesis gives us this other idea, which is that virulence will evolve to a balance between the host living for long enough to keep spreading, but there being enough virus at any given time that the virus can spread. It's been really quite difficult to observe that in any infections. Probably by far the best example we have of actually precisely that happening is when myxomatosis was introduced to control rabbits in Australia. Again, another fantastic story, but not a story for today. Actually, what did happen was that myxomatosis did evolve to intermediate virulence. Oh, and Hamish is in the audience, so he could tell us if we wanted to talk about that. And in HIV, the story has been, the data has been pretty mixed. So there's been there's a study that's of widespread interest, but you could probably take your pick. It's one of those bits of biology where there are many, many different publications, and you could pick your one, whichever answer you wanted to give. I'm going to show you my favourite one. And this was work that was done with Philip Goulder in the medical school here, and my part of it was, well, I was going to say done with, actually it was done by Hannah Roberts, who's a graduate student of mine. So the idea that Philip had was that he was going to compare Botswana here with South Africa here. And the reason for doing that was that, roughly speaking, the epidemic in Botswana is about 10 years behind the epidemic in South Africa. So here's time in years along this axis. Here in blue is the South African epidemic, and here in turquoise is the Botswana epidemic. I think I may have said this the wrong way round, sorry. The Botswana epidemic is 10 years ahead of the South Africa epidemic. And you see there's about a 10-year gap there. So this was a way of saying, well, let's see what's the difference between the way this epidemic is behaving in this country, Botswana, and this country, South Africa. So with large numbers of samples, so here are the numbers of individuals here, 15,000 from Botswana, 29,000 from South Africa. Philip and his team went on to do a big natural history study. In Botswana, so here's Botswana compared with Africa, and this here that says viral load basically says if you took a little vial of blood out of one of these patients' arm and counted how much virus is in one drop of their blood, how much is there? And what you see is that the viral load is a little bit lower in Botswana than it is in South Africa, but with these huge sample sizes, you can say very clearly that it is clearly statistically significantly lower. And equally, you can say in Botswana, the CD4 count, so that's a measure of how poorly the person is. The lower the CD4 count is, the more advanced disease has become, and the more sick you would expect the patient to be. You would say in Botswana, these patients have lower CD4 counts than in South Africa. And that's sort of what we would expect from an older epidemic, because more of the people in Botswana have been infected for longer. So then if we ask the question, if we take virus out of people and we very carefully grow it in vitro, we're going to take it into the lab and grow it, and very carefully count how many viruses there are each day, and calculate how fast each individual patient's virus grows, and then compare it with a virus that we know a lot about, what do we see? Okay, so that's what viral replicative capacity is. So that's an in vitro measure. In vitro means in glass. So it's a laboratory measure of how fast does virus grow from patients in Gaborone, that's in Botswana, or in Durban, that's in South Africa. And what we see is that there's this small but statistically significant difference, so that the Gaborone patient's virus is a little bit less aggressive when you grow it in vitro than the Durban patient's virus. And if we look at data just for, so these green dots now are that data spread out, what you see is that the patients who have virus that grows more quickly tend to be the patients who are more poorly, have a lower CD4 count. So rapidly growing viruses that grow fast in vitro, are slightly associated with patients who have lower CD4 counts. So this picture here, because this epidemic is older than that epidemic, we would take as evidence that HIV in this part of southern Africa is indeed becoming less virulent. There are two possible explanations for what's going on here. So one is that these viruses have been in this population for longer, and they've been under longer assault from the immune responses of these patients, and those immune responses have selected viruses that are less fit. I'm going to talk in more detail about that in a moment, okay? So one possible explanation for this difference is immune-driven viral evolution. And another possible explanation is that there's been more treatment, basically treatment rollout was earlier in Botswana than it has been in South Africa for reasons that many of you will know. And that when you treat people for HIV infection, what you've tended to do, at least in the past, is to give these very effective drugs to patients whose CD4 count has become low. So to patients who you are worried are going to become sick soon. And you can see from this graph here that if you selectively treat patients who have a low CD4 count, on average, you're more likely to be treating a patient with a highly virulent virus. And that's a way of exerting a selection pressure that favours these viruses here, the ones that make patients sick more slowly. Nevertheless, one has to admit that this difference is quite small, and we ask the question, the very obvious question, well, if you can see it here, why has this been so difficult to see elsewhere? Because this is not the place in the world where the rollout of the treatment of HIV has been, was certainly not the most successful. And that's where Hannah, with some help from me, came in. So Hannah made a mathematical model that consists of a set of ordinary differential equations that ask the question, would we expect treatment, widespread treatment with antiretroviral drugs, to speed the evolution of avirulence? So what we do in a situation like that is we make a model of infection in which we say, well, let's suppose that there are 4 sorts of infected people. You can be infected with one of 2 viruses, one that makes you sick fast, one that makes you sick slow, and you can either get treated or not. And we set things up so that indeed people with the virus that makes you sick fast get treated sooner. Because that's the question we want to ask is, does treating people sooner who have virulent viruses favour avirulent viruses? And what Hannah found is what you would expect, namely that yes, it does. But she found another couple of things along the way that I think are quite interesting and perhaps help to understand why this picture has been so confused. So here's a mathematical model just expressed as a set of boxes and circles and arrows here. It could be written down as a set of, that one could be written down as a set of ordinary differential equations. And here are some numerical simulations of those equations. So there's time in years along here. This first picture here is the number of infections if you don't do any treatment, okay? And what you see is that the highly virulent virus is the one that spreads fastest. And that's because, remember, we've made that assumption that something that is very virulent is also very transmissible. We said there's a trade-off. So a virus that's very virulent makes the patient sick quickly, does that by having a lot of virus all over the patient, so that patient is also going to be more infectious per unit time. Okay, so the first thing that happens is that you see a lot of spread of the highly virulent virus. And let me tell you that it's not because there's enormous difference between these two. The transmissibility of these two is probably less than 10%, but you see this enormous difference because they're in competition with each other, fundamentally. If you start treating, you still see the most virulent one. Actually, you start treating here, what you see is two things. First of all, long-term, yes, you're favouring the blue one here, the less virulent virus, but also, because the population infected with the highly virulent virus turns over more quickly, unfortunately those are people who in this model are dying more quickly, that population responds much faster. So actually, for a new reason, not the one we originally thought of, you figure out, yes, I would expect treatment to favour avirulent viruses. But the other thing you find out is that I don't expect this difference to be extremely large. And that's one possible explanation for why this hasn't been something that's been staring us in the face the world over. And I think another explanation is this pattern here. Sorry, so this is the same time in years along this axis, and this is the proportion of infections that are with the most virulent strain. So this is a surrogate for what's the average virulence. And what you see is that even without treatment, this is not a monotonic curve. It doesn't always go up or always go down. And we look at this picture, so look at the solid line here, and we say, well, maybe the reason some people find when they view these studies that virulence in the population they're looking at is increasing, other people find that virulence is decreasing, and others find no difference, is that they're all looking at different sub-epidemics, and they've happened to be looking at different points in this curve. So for me, actually, the outcome of this study that is most interesting is that this is really because of the very early spread of the more virulent strain, we wouldn't expect temporal trends in virulence to be monotonic. We'd expect these much more complicated patterns. And that helps me, at least, to understand why this has been so difficult to unpick in HIV infection. And then the other question that Hannah went on to ask, and I'll just show you this, is why was the effect so small? So in the modelling, the effect was very small. And it boils down to the fact that although our intuition is mostly right, it misses out on the real subtlety, which is that what matters is not how many people you remove who've got a highly virulent infection, but what proportion of the time that people are around with highly virulent infections you're removing. So that's something that Hannah was able to thrash out by making a partial differential equation model. It's a bit more complicated, and I'm not going to go into it now, but I think what Hannah's done with this modelling is to really extend one's intuition from the thing that you just sort of think is obvious into an understanding of why some of these effects are really quite subtle, which again explains why they've been quite difficult to see. Right, that brings me to question number 2. How fast does HIV evolve within infected people? So I said, sort of slightly quickly, that one of the explanations for the evolution of avirulence that was being seen in that South African comparison might be because of immune-driven evolution. And I now want to talk a little bit in more detail about what I mean by immune-driven evolution. And before I do that, I must remember to tell you that what I'm going to talk about now is work that was done in collaboration with Rodney Phillips, who used to be my co-director at the Institute for Emerging Infections, and also with John Frater, who is my current co-director of the Institute for Emerging Infections. So the mutations that were being tracked in Philip Goulder's paper were what's called CTL escape mutants. And I'm going to show you some pictures now of what those are. And to do that, we have to do a little bit of mathematical modeller's immunology, which consists of circles and simple shapes. Imagine that that's an HIV-infected cell, and that's something called a cytotoxic T lymphocyte. It's a kind of white blood cell, and this cell's job is to figure out that that cell is infected with something that didn't come from this cell's body, and then kill it. And the way it does that is the following. a small part of virus protein, really a very small part, about 9 or 10 amino acids long, is loaded onto a little bit of self-protein and shoved out onto the surface of their cell. Our own bodies do this all the time, and most of our cells have this kind of protein in them. We're able to do it. This little part of the virus that the CTL, that the immune cell ought to be able to see, is called an epitope. So what happens is that the CTL comes along, recognizes that that's non-self, kills the infected cell. Job done, fantastic. Notice that I'm going to go backwards one, was an extremely specific interaction. And also, it was an interaction between this piece of immune machinery, effectively, and a small linear chunk of virus. So this CTL doesn't see whole virus in all its sort of folded up glory. It sees these short, cut out pieces of virus that are, as I said, just 9, 10, 11 amino acids long. And I'm emphasizing that now because I want to come back to it later. I think it helps explain why some of these explanations really work quite well. So that's how it should work. Infected cell dead, job done. But many viruses, many pathogens in fact, HIV included, evade such immunity. And they do that simply by throwing out mutations at random, some of which will sometimes fall into an epitope, so a bit that the host cell ought to see, so that the immune cell ought to see, and then the immune cell comes along and goes, don't recognize that, because this extremely specific interaction no longer works because there's been a mutation at this one site here. So that's what I mean by CTL escape. There's another really important thing, which is that different people have different HLA molecules. So if I caught flu, let's say, let's be more cheerful, my HLA molecules will show my body different bits of flu virus than if my husband catches flu. Okay, and that's really, really important. So we're going to say, we call a little bit of virus that person's that person's HLA can present, we call that HLA matched. Okay, so we're all HLA matched for some of the viruses that are in us, but not the same ones. Okay, and that's going to be very important for this bit of this story. Okay, so different people make responses to different bits. Sorry, I've got myself in a muddle here. Make responses to different bits of viruses. No, I don't want to go to that. Right. We have all, particularly here in Oxford actually, been very interested in this phenomenon of CTL escape for a long time. It was first described here in Oxford by Rodney Phillips when he was a postdoc working up at the hospital. But really, I think the reason that we're particularly interested in this phenomenon is that it gives some data that allows us to work out how strong different bits of the immune response are. And that's not something that's easy to do. We have lots of fantastic technology that lets us work out how big bits of the immune response are, so by counting cells or counting soluble factors and things like that. But actually, from the point of view of working out, well, yes, but what are they doing? So if you're making a response to that cell, how much faster do cells infected with that virus get killed? Because that's really what we'd like to know. And we've used, here in Oxford actually, we use data on how fast inside infected individuals these kinds of viruses grew out to work out how strong is that particular immune response in that person. But I have to take a step back first. So first of all, at the stage at which CTL escape mutants were being described quite widely in the literature, what would happen is that somebody would do these fantastically detailed studies where you take a patient and you take samples from them let's say, every 10 days for a couple of hundred days, say 6 months or something, and then you sequence the virus in that patient and you work out at what rate is the escaped virus growing. Because that's what happens, is that these mutations arise and then they grow out because that particular virus then has a fitness benefit because one bit of the immune response can't see it. And there's absolutely beautiful natural history published by people from around the world describing that phenomenon. And what they sometimes say in those papers is, my goodness, look how fast this is. So it tends to happen in something like 100 days, so 3-4 months. And if you know that an HIV-infected cell only lives for a day, and you say, well, that means it's taking 100 generations for this more-fit virus to go from effectively absent to effectively the only thing there, then you start to think, hang on a minute, that's actually quite a small fitness advantage. If it takes you 100 generations to go from absence to fixation, and we know that compound interest makes these things happen faster, then your fitness advantage is less than 1%. Now, a 1% fitness advantage, well, the geneticists in the audience will think, oh, well, that's quite big. But actually, from the point of view of the ecology of what's going on here, the fact that a cell that can't be seen by one whole chunk of the immune response gets a 1% advantage tells you that that immune response is not doing a huge amount. So then all the CTL escape people said, oh, well, hold on a minute. We have some other patients who we've looked at much, much earlier. So all those patients we looked at before, they were all people who've been infected for years and years and years. Here are some patients, and when I say some patients, I mean 3 or maybe 4, who we're looking at just in the very first weeks that they got infected. And look, the rate at which the escape variants grow out is much, much faster. And some of us then said, well, yeah, but that's 3 or 4 patients. Is that normal? So Rodney and John did the thing that you need to do, put together the gold standard study, which is a cohort study. So you find 100 people who've been quite recently infected, and then you just followed them. Take blood from them every 6 months, and you say, how many new escape mutations am I seeing in these people? And the way you display data like this, again, this is an analysis that Hannah Roberts has done with some help from me, is in what we call a Kaplan-Meier plot. So here we've got 2 years of data, so going from 0 to 100 weeks, just concentrate on the left-hand side for the moment, and this is proportion of patients with no escape. Okay, so this is the proportion of patients who've had no new escape event happening in them. And what we thought we would see from what had been described about these very rapid, very early events in a handful of patients, this would plummet like a stone. And it's not. So after 2 years of being followed, these patients, it's something like a third of these patients have still not seen a single new escape mutation. We can split them out. We can also look at patients with different sorts of immune response, but that's less important for now. So this is really a very surprising result. This is much, much less immune escape than you would have thought from the state of play before this study was done. And it raises a series. The first question you could ask is, well, we know that on average, these patients were seen something like an average of 12 weeks after they got infected. Maybe we missed it all. Maybe everything that happened was over before this study ever began. But we can address that by asking the following. So again, look at the left-hand picture. Now, this is a really complicated picture. So the first thing I want you to notice with the left-hand picture is that most of the dots are near to the line x equals y. Yeah? Most dots are near this straight line here. So what we've got here is, at the very first time that these patients were seen, what proportion of patients who could make an immune response had an escape in each different epitope. So each one of these dots is a different little stretch of HIV. So here's a famous one, for example. Here's Taftipsy. So in this cohort, I'm pretty sure there were 9 patients who had the right kind of immune response. They had the right HLA, so that they could have made a response to to Taftipsy. And then there were also a large number, there'd be more like 60 or 70, who couldn't. If what we'd seen was a huge amount of escape driven by immunity that we'd missed, so that had happened before we took the first bleed from these patients, all these dots ought to be over here. Because the patients who can make a response should have driven an escape. And what you see is that, yes, that is true for a couple of them. Most of these are restricted by extremely rare HLA, so these will only be one patient. But for the vast majority of epitopes, what we see is that the prevalence of escape is high. There's lots of escape here at first sample, but it's pretty much the same whether you're making a response or can't make a response. And what that says to us is that, yes, there's a lot of escape in these patients, but it was transmitted. These patients caught these escape variants from other patients. This is not some reflection. This isn't what you'd expect to see with within-host evolution. And a year later, you do actually see some within-host evolution has happened, but as I just showed you, it's not a huge amount. Then the second question we asked was, well, maybe there's some subset of patients who are very escaping. Maybe most patients do not very much, but there's some small subset of patients in whom there are lots and lots of escapes. Well, that's very easy to test because we can just count up the number of escape events and compare it with what we would expect just by a Poisson distribution, and what you see is there's really no difference. It is not the case that there's some small subset of patients in our cohort who have an awful lot of escapes and most have none. The distribution is just exactly what we would expect at random. So that leaves us saying that for this kind of evolution, within-host evolution is slow. Now, of course, at the population level, these variants are clearly accumulating. There's a lot of escape out there, but we would say most of it is transmitted from one patient to another, not driven by the immune response of the patient they're in. How am I doing? Third question relates to these same escape mutations, CTL escape mutations. So we've been measuring how fast these things happen, mostly based on data from Switzerland. And we'd really like to know, do we see the same pattern if we look in other places? Because we're very interested in escape rates as a way of characterizing different viral mutations, but that's really only meaningful if we see the same pattern when we look in different places. And this is a piece of work that was done with my then graduate student, Duncan Palmer, who was co-supervised by Gil McLean. So what we're going to do here is quickly compare data from 3 places now. So from a big study in Switzerland, a second big study in British Columbia, and a third in South Africa. So these are 3 sub-epidemics. What we want to know is, if we estimate these escape rates, do we get the same answer if we look in people in different sub-epidemics around the world? Unfortunately, we don't have the same sort of beautiful cohort data as we had before. So it's not a case of just saying, well, I'm just going to count up how many escape mutations I see as time goes through. And so instead, what Duncan had to do was make a much more ingenious way of inferring escape rates from cross-sectional studies. So now what we have is a single set of viral sequences from patients at a single point in time, but we do know which patients can make an immune response to which parts of that virus. And to cut a long story short, really quite surprisingly, we find that escape rates are the same in different places. That's not so fantastically surprising if I compare Switzerland and, say, Switzerland and British Columbia, but comparing viral evolution in that detail with the sub-epidemic that's happening in South Africa, with a sub-epidemic that's been running in Western Europe, it was really quite a surprise to me that they should be so similar. Even more surprising was when Duncan dug out a similar set of measures of how fast can these viruses grow in vitro in the laboratory. Now what we would expect to see is the opposite pattern. We'd expect to see that a virus that can grow very fast in the laboratory, so with high replicative fitness, would have a low value here, because this is the log of the reciprocal of the escape rate. So if something escapes fast, get this right, if something escapes fast, that will be a small number, because that's the waiting time till you escape. And indeed, that is what we see. So we see here a highly significant negative correlation between these two things. So this is something that was measured with single viruses in a test tube in a laboratory. And this is something that was inferred from a whole population of people living a real life and actually transmitting these viruses between each other. before I'd done biology for lots of years, I started life as a mathematician, I would have said, meh, yeah, what did you expect? But I've done enough biology now to be really surprised by this. And I think the reason that this works as well as it does goes back to the issue I was talking about, that we're looking at this fantastically specific response on a small linear piece of viral DNA. So a lot of the complexities of protein folding and things like that are taken away for us. So to summarize the work that Duncan's been doing, what we find is for escape rate, so that's the top half of this picture, for the 3 places that we looked at, actually there are 4 places, but we can leave them out, we always find strong correlations, well, in particular we find statistically significant correlations. A black dot means that correlation was significant at the 0.05 level between escape rates. So we just see the same escape rates across 20, 30 different epitopes again and again and again. And more surprisingly, I think, we see these statistically significant negative correlations between escape rate and in vitro measures. The bottom half of this picture, that is not nearly so clear, is what happens with reversion rates, and I'm not going to go into that. I come to my 4th question, which was, does HIV still evolve in successfully treated patients? Oops, sorry. So to talk about this, I need to show you, first of all, some published data about what happens when an individual patient is put on what we call highly, is that what they call it here? Well, it's put on extremely good therapy, let's call it. So this is months along this axis here. Each line is a different patient. And this is an absolutely standard picture that you're, and this axis here is that same thing again, how many copies of virus in a drop of blood. And this is a standard picture that what you see is that in the first, straight away, in the first week or so of treatment, you see this enormous drop in how much virus there is in most patients, not this one here, notice. And that then slows down, how fast that drops. And then in most patients, eventually it goes to, it falls below the level of detection. And this is data showing you how amazingly successful these drugs are. Unfortunately, with the great majority of these patients, if they stop taking the drugs, this virus will be back to within half a log of where it was before you started treating them within a couple of weeks. So these patients haven't been cured. This is called suppressive therapy. So they're on a treatment that keeps their virus under control. They will be much, much healthier and virtually uninfectious to other people. But if they stop taking the drugs, this virus will grow straight back. And a big question in the world of HIV infection is what's going on here. So the virus in their blood is below the level of what we can detect, so it's quite difficult to study. But we know that somewhere inside these patients there is a reservoir of virus that once they stop taking drug will come roaring back. And the received wisdom is that reservoir of virus is cells that have become infected, have laid down viral RNA as DNA, and are in a resting state. Remember that HIV infects cells of your immune response, some of those cells being cells that mean that, for example, I'm never going to catch measles again. I had measles before I was 1, and I'm immune to measles. And that's a great place to hide if you're a virus, is to hide in a long-lived, quiescent cell whose job is just to sit there in my body waiting for measles to try and get back in me, because that's how immunity works. So we'd really like to know, is that reservoir maintained just by sleeping cells, or somewhere in these people are there small sanctuaries where small amounts of virus replication continue. So this is a second set of data from the same patients, and this is about intracellular antiretroviral concentration. So this is about how much drug is there in different places in these patients. And this is a log scale. And what you need to see is that whilst there's plenty of drugs in the cells that live in their blood, there's, for many of the drugs, not plenty, several orders of magnitude, 100-fold less in their lymph nodes. Now, I'm going to do something really annoying. and not tell you what we found. And instead, I'm going to show you these beautiful pictures of the laboratory where all this work was done. The reason I cannot tell you the results of this study is that they're under review in a high-impact paper. If I show them here on a broadcast talk, I could completely destroy the chances of publication of that work. So come to the pub if you want to hear, but right here, right now, I can't show you the results. What I can tell you is that we think they're extremely exciting and have profound implications for the way HIV is treated. So let me finish. I've set out to address 4 questions about HIV evolution. Those were, is HIV evolving to become less virulent? We think yes. How fast does HIV evolve within infected people? We say slowly. Are patterns of HIV evolution consistent across epidemics? That was in 3 different places. Yes, we think astonishingly so. And does HIV still evolve in successfully treated patients? Invite me back next time. I just finished by thanking the people who really did this work. And the 3 most important of those for me are Hannah Roberts, whose name I've misspelled. I hope she's not here. I haven't got my glasses on, so I can't see. That's Hannah Roberts, with an S is her name. Who's funded by the MRC, Duncan Palmer, funded by the EPSRC, and Helen Fryer, funded by the Oxford Martin School. And then also very important to Philip Goulder, Rodney Phillips, John Frater, Gil McLean, and Steve Walensky, who are all the people we have worked together with in order to do these studies. Thank you.
Please don't ask Angela what the results of that last piece of research are. We will do a news release on it with the university in good time. But please do ask Angela other questions. This is being live webcast and recorded, so if you don't want to be on the internet or on video, I'm afraid get somebody else to ask the question for you. We have a microphone, so I would ask that you wait for it to arrive. Who wants to go first? Silence. Okay. Sorry about that, it's very unusual. Oh, we have one at the front. Everybody's being shy.
We talked about the HLAs being different in different people and obviously presenting different epitopes. Does that influence greatly the general results? And is that an area of research that is being looked into?
The general results of infection? Yes. So the biggest single predictor of how fast you're going to get ill if you get infected with HIV is how much virus is in your blood. But the biggest single indicator that's really about you is your HLA type. It's not an enormously strong effect, but it is a statistically significant effect. So you'll have seen there was a slide that I didn't really go into that talked about protective HLAs and not protective HLAs. So for HIV, which I think is probably the best understood of these, we know that certain HLA types tend to do better. It's not rock solid. It's not that they always do better, but on average they will do better.
I've got a question if there isn't anybody else picking their hand up. I'm just wondering what lessons can be learned for other types of virus from some of these questions that you've posed and answered of HIV.
Yes, I'm inclined to give a very sort of Oxfordy answer to that, which is big, simple studies. So big studies that have enough people in them and get the right samples. Actually, that's what I was slightly rushing. I was going to do a great big advert about, so you have to have the samples from the right place, in the right quantity, and of the right quality. So, for example, I started off by saying, well, look, there's this great big ding-dong going on with Ebola, and it's fundamentally boring because it's not going to be answerable. There was a pretty long ding-dong that went on about, well, how fast do these CTL escape mutants arise, until John Rodney gathered a cohort study and did the right study. And that took years. And it actually was, it was done on the back of a clinical study to ask a different set of questions. You'd never be able to fund a study like that to ask a sort of purely scientific question. But what you can do, what Rodney did was teamed up with a big group run by John Weber at Imperial, who were doing a study about different ways of treating people in order to do the basic science as well.
I think there's an interesting point there for people and how they look for funding, approaching things from a public health point of view in order to look at.
And I mean, I think another lesson I would say is that there are some questions that only require a small number of patients, but there are other questions that answering with a small number of patients isn't an answer.
Anybody else? Oh, right. So here and then at the back.
Thank you very much for an interesting talk. Could I just ask you what happens in the immune privileged areas of the body and how that affects the viruses that sit in there? I was reading about it following Ebola, so it's been in the press quite a lot. So just in the context of other viruses.
Yes. There are immune privileged areas. Presumably you were reading about this in the context of the nurse who so happily has gone home today. I'm going to pass this to my colleague who's visiting from Chicago and is actually a physician. Steve Valinsky, tell us about immune privileged areas.
Thank you, Angela.
You're welcome.
Excuse the American accent. Yes, indeed, there are certain portions of the body in which different organisms may grow without having any recognition by the immune surveillance mechanisms that exist. Certainly that we've seen in Ebola patients, it has been within the vitreous of the eye. So we've actually seen that the virus, even though we don't detect it out in the peripheral blood, is growing quite nicely inside the eye. And there's been reports of some of the people that have had this infection of the eye color changing and been associated with the fact of that. And we've seen this with other viruses and other immune privileged sites as well, that it's able to still replicate even without us being able to detect it within the blood, but yet remain within the tissues of the individual. So it's an interesting question, and it's a conundrum of why it exists, what we can do about it, and how do we follow it, and what we can do in order to actually cure an infection. So it's quite an interesting question. So, and since you had one.
Angela, I was intrigued by the similarity of the rates of evolution in different parts of the world. Can you speculate about why that's the case?
Yeah, so my speculation is that that boils down to the intimacy, the specificity of the interaction, and the fact that it's an interaction just on those 9-mers. So these viruses are very different, but because it's just that bit of your immune response only looks at these very small, short pieces of viral antigen. So it would appear that epigenetic interactions are either always the same or they're not important.
Any other questions? I feel like an auctioneer. If that's all we have, I haven't got any from Twitter today. Before we thank Angela for what I think was a really in-depth and fascinating look at the cutting-edge research that's going on into the HIV virus, could I just take a moment to flag up a couple of forthcoming events as well? Next Thursday's seminar at the same time here is Julian Savulescu, who is going to look at the ethics of enhancement, so posing the question, can you create the perfect human being? On the following Thursday the 26th, a seminar which I think follows on very well from today's. It's Professor Adrian Hill, who's looking at the issue of prevent and protect vaccines and immune responses. So I think it's the perfect follow-on to today's. Those of you who've come to a few events will know that on the 23rd of November in the Sheldonian, we're very privileged to have Lord Martin Rees giving our 10th anniversary lecture. And he will address, will technology's dark side eclipse humanity's bright future? If you've never heard Lord Rees speak, I really would urge you to come to that. It's going to be a wonderful, wonderful event. And then on the next evening, the 24th of November, back here, the Institute for New Economic Thinking has Lord Adair Turner launching his latest book, and he has a fascinating insight into the real causes of the 2008 financial crash. So obviously not a follow-on to today's, but equally fascinating. It's been described by George Soros as an eye-opening book, so I highly recommend that as well. So with that plug for our events, I would also like to plug, if you haven't already seen it, our 10th anniversary report. Angela has an article in it. There are plenty of copies, so please do help yourself as you leave. Thank you very much for coming, and please do come back. And thank you, Angela. That was brilliant.